All right, guys. So in this video, I'm going to be going over numbers 2 and number 4 on the FRQs that I have posted for this week. So this first one that I'm going to go over on number 2 is about photosynthesis. So let's take a look at what it's asking about, what you should be able to answer based off of this. So the passage says that an absorption spectrum indicates the relative amount of light absorbed across a range of wavelengths. The graphs above represent the absorption spectra of individual pigments isolated from two different organisms. One of the pigments is chlorophyll A, which is commonly found in green plants. The other pigment is Bacteriophytopsin. So that's another species, and it's commonly found in purple photosynthetic bacteria. The table shows the approximate ranges of wavelengths of different colors in the visible light spectrum. So graph 1, it looks like we have some absorbance right here around 550, 575, somewhere around there. And this is showing you that it is able to absorb this amount of wavelength. And in graph number 2, it looks like we get some absorption right around here, nothing around the 5 to 600, and then it spikes a little after, like, I'd say maybe around 660. So, excuse me, it tells you the color matching up with the wavelength. So this basically tells you, if you don't remember how to read these things, this color that it's actually able to absorb. Right here, if we say that this is around the 500 wavelength, this would actually be the color green because it can actually absorb that color. So the absorption here is right around the 550 to maybe 575 wavelength that's absorbing green. So, and then if you look back over here, we get a nice peak right around, not quite 450, so I'll say that this is around 430. So somewhere in that realm. So like right here, this one would be absorbing purple. And then right here, right around 650, this would be able to absorb red. So basically this means that this graph is showing you that whatever plant this is, whether it's the chlorophyll A or the Bacteriophytopsin, you're just having to figure out the color that they can absorb. So this is what they're giving you. So let's go ahead and see what the question is based off of. So part A, explain why measuring light absorbance is a good indication of the amount of photosynthesis occurring. So this one is pretty simple, but it is worth your time to revisit kind of what's going on here. So this just shows you that plants, this is just taken from a stock photo on the internet, right? Where plants have more than one type of pigment. Excuse me. And so chlorophyll A, just if you look here, chlorophyll A, this dark green one, can absorb the wavelengths right around, let's say maybe 430, and then that dark green peaks around maybe 670. And then you also have chlorophyll B, which is absorbing this wavelength right around maybe 460, 470, and then it also spikes right around 650. And you have a carotenoid pigment, which is in yellow. It absorbs right around maybe 460 or so, and then right around 500, but it doesn't spike over here. And then you have this third pigment, or excuse me, fourth pigment, chlorophyll F, that spikes right at a little bit past 700. So let's talk about and answer that question. Why does it matter? Well, if you forgot, these things in green, this is right, the light reaction. These little things in green, those are the pigments. And so the reason why we care about those pigments is that the more light that they can absorb, the more energy it's going to have, and that makes it easier to actually split water, pull the electrons off of water. So this is just showing you that all that light energy, these are all the pigments, they can actually absorb that energy, pulling electrons off of water, right? So just kind of while we're here, we can revisit. Remember, the electrons go from photosystem 2 to photosystem 1. The electrons get excited again, and then they end up on NADP, making NADPH, right? And just to remind you that this goes again to the Kelvin cycle. So the key thing here is that why does it matter? Well, remember, the more light it can absorb, the more energy it's going to have, and the easier it's going to be doing photosynthesis. So, and just understand that each one of these are different pigments, and these two photosystems have different absorption spectra. So this one absorbs right around 680, this one absorbs right around 700. And that has to deal with the type of pigments that they have. So the best and simplest answer for this would be, why is measuring light a good indication of the amount of photosynthesis occurring? Again, the more light absorbed, the more light absorbed is going to have an increase in the light reactions, which will eventually increase Kelvin cycle. So something along those lines that as long as you're mentioning that you understand that the more light absorbed, the more efficient it's going to be at doing the light reaction. So understanding that, and this is just showing you another emphasis on this, right? So if you have any questions on that or you're wanting to revisit that, go back and look at my chapter 10 video notes. So let's look at part B. Identify the experimental question posed by the researchers, justify the need to measure across the entire visible light spectrum. So I would just say in terms of this, let's kind of go back and see what the question was about. What do they actually do? The graphs represent individual pigments isolated from two different organisms. One of the pigments is chlorophyll A. The other pigment is bacteriophytopsin. And this just shows the amount of wavelengths that they actually have. So let's kind of do a quick analysis of what we were measuring. So notice that they were given all the same amount of light. So what's the variables here? I would say that the variables here in this is going to be the type of pigments that they have, and they're measuring the absorbance. So in terms of this, my question would be, do all pigments absorb the same amount? Okay, that's kind of my question, the experimental question. Justify the need to measure across the entire visible spectrum of light. So just understand this in terms of this, if this was sunlight, remember that the sun produces all wavelengths on this one. So that would be my justification, is that the sunlight produces / gives all the wavelengths of light. So that's what I would emphasize on those. So that's basically the question. And that one was kind of sneaky because in terms of this, you're not measuring photosynthesis in this, you're just measuring the amount of absorbance, which eventually will correlate to the amount of photosynthesis is doing. But in this case, we had two species and we gave all wavelengths of light. And in terms of this, let's go back to this one. How do you justify the need to measure across the entire visible spectrum? Again, sunlight produces all that, but another thing too is that each pigment is specific. Each pigment is going to be specific to the wavelength it absorbs. So I would even add that to this as well, that each pigment is specific to the wavelength it absorbs. All right. Okay, moving on. C, identify the pigment used to generate the absorption spectrum in each of the graphs above. Using evidence from the data, justify your response. Okay, so basically we got to figure out which was this one and which was this one. So we had two things to pick from. One of the pigments, chlorophyll A, it tells you is found in green plants. The other pigment is bacteriophytopsin, and that's found in purple photosynthetic bacteria. Okay, so remember, if it is not absorbing it, it's reflecting it, and that's the color that it's going to be appearing. So that is something that you just wanted to make sure that you're able to kind of process here. So in this case, right here, let's look at this graph one. It's actually able to absorb this wavelength right around a little over 600. So in terms of this, or excuse me, not quite at 600, right around 550, 550 to maybe close to 600. So this kind of falls within that realm of that 495 to 570. So this can actually absorb green light, right? It can absorb it. Now notice the colors that it's reflecting. It's all kind of right here in this realm. So right between 400 and 500. And that's like your blues, your violets, your cyans, and all that stuff. So the fact that this is being reflected if it's reflecting the color, that's what you would actually see. So in terms of graph 1, I will write this off to the side. Graph 1 is going to be the bacteria Phodopsin. And then graph 2 is essentially the chlorophyll A. And again, the reason why I know that this one is going to be green is because notice it's not absorbing this these colors right over in between the 5 to 600. And so if you look here, the 5 to 600 realm, these are going to be like your greens and yellows. So this actually would be green in terms of what you would actually see. So using the evidence of the data justify response, you would just have to explain that if it absorbs it's not actually going to be that color. Whatever it's unable to absorb, that's the color that you would actually be able to see. So that's how you would justify your response on that one. So moving on to the last one for this question, for part D. It says, in an experiment, identical organisms containing the pigment from graph 2 as the predominant light capturing pigment are separated into 3 groups. The organisms in each group are illuminated with a light of a single wavelength, 650 nanometers for the first one, 550 for the second, and 430 for the third. So we got 3 groups we got to worry about. The 3 light sources are of equal intensity and all organisms are illuminated for equal lengths of time. So basically think about it, you have basically 3 light bulbs. I'll do a little light bulb here. I'll do another really poorly drawn light bulb here, and then another poorly drawn light bulb here. But the point is that this group, one of them is giving off 650 nanometers of light. This light is going to be given off 550. And this last one is going to be 430 nanometers. So basically you have the plant and it has this pigment here. This is, we identified this earlier, is that this one is chlorophyll A, okay, according to that last one. So we have your plants and they were illuminated and separated into 3 groups. Okay, that's what it's telling you. This light is giving off this color, this light is giving off this color, and this light is giving off this color. So now we're going to put this into context to figure out, okay, so what's going to happen here? What's going to have the maximum amount of growth? Predict the relative rate of photosynthesis in each. Justify your prediction, okay? So I'm going to call this group 1, I'm going to call this one group 2, and I'm going to call this group 3. So if you look through this at group number 1, group 1 is going to give off 650 nanometers of light, okay? So I'll just label this one right here as group 1 on my graph, okay? Group number 2 is given 550 nanometers of light. So right around here, that's where my group 2 is going to fall. And then if you look over here, 430 is right around here. That's where your group 3 is going to be, okay? So basically you have to put this into context. And let's start with the lowest rate of photosynthesis. So I would say that group number 1 is going to have the lowest because it's not absorbing anything. That's your justification. It's not able to absorb that wavelength. And then you have to just figure out, well, what's going to be higher, group 3 or group 1? Group 3 is actually going to be the highest rate. And this is going to be the, I'll just say medium amount. But it can still absorb it and do some growth. So the highest group of photosynthesis activity would be group 3. And your justification is just as simple as it can absorb the most light. So the more light it absorbs, the better it's going to be at photosynthesis. Group number 1 with the 650 nanometers, it can absorb, but not quite as much as that third group. And group 2 is going to be the lowest because it can't absorb any of that light. So there would essentially be no growth. And that's your justification. So that's what you would want to make sure that you're able to explain in this one. Yeah, that was a pretty well thought out FRQ by them, and kind of testing primarily photosynthesis, a little bit of scientific method. So let's move on now to number 4. Number 4 is going to be, this was given, I remember 2018 is where this one was from. And it's kind of interesting. So seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce a pollen that is carried by circulating water to female flowers, resulting in fertilization. So basically it's showing you that, or explaining that we're looking at two flowers, the male and the female flower. A researcher claims that mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of circulating water. To investigate this claim, researchers set up aquariums to model the possible interactions between the invertebrates and seagrasses. Use the symbols below and the template to demonstrate the experimental design for testing. The researchers claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Draw appropriate symbols in the negative control aquarium and the experiment. Don't use any symbols more than once in the same aquarium. Okay, so first and foremost, let's kind of identify what's going on here. So if you don't know what a negative control group is, a negative control just basically is, it's a control group, but when we say it's a negative control, you basically don't expect any results. So basically it's a control group, and not only is it a control group without your variable, it's a control group where you're not expecting any results. So if it asks you like what's a positive control, that would be where you would set it up without the variable, but you're still going to be expecting results. So look, if you're testing a drug that would essentially stop a process from happening, that would be your experimental. Your positive control would be something along the lines where it would be your control group, but you're still expecting to get a result. So just wanted to point that out if you don't know what a negative control is versus a positive control. So just basically think of it as a control group. It's without your variable, and you're not expecting any results. So let's figure this one out. So we have the male and the female flower. What would be an appropriate control group? It's as simple as this. So on our control group, I would just have the male flower and the female flower. That's it. So this just means that you have these two flowers with no invertebrate. The experimental one, you have the male and the female flower, and you're also going to be adding the invertebrate because that's what they're claiming is going to pollinate these flowers. So really weird drawing on this one that you had to do, but that's essentially all. That's it. So remember that that invertebrate is the variable. That's what they're saying will yield the fertilization. Okay. So let's move to B. B says identify the dependent variable, so I'll write B over here. The dependent variable in the experiment. So what are they actually measuring? And it tells you right here earlier in the passage. So the dependent variable is actually fertilization. Okay, that's what they're measuring. Predict the experimental result that would support the researcher's claim that mobile aquatic invertebrates can also transfer pollen from male to female flowers in the absence of water. So basically, how would they justify? Well, what are they expecting to see in the experimental aquarium? In the experiment aquarium, just an increase in fertilization. That's about all that they're going to be measuring. So that was an easy 4.1, just really more on the scientific method. And so those were FRQs numbers 1 and 4. So, or excuse me, 2 and 4. And we'll see you guys in the next one.